Hello everyone. This video will be discussing on the importance of mammalian downstream technologies in tackling COVID-19. The outbreak of coronavirus disease, COVID-19, which started in Wuhan City, China, is an infectious disease caused by a newly discovered coronavirus. The pandemic has sickened more than 100 million people worldwide and over 2 million people have died because of COVID-19. The emerging COVID-19 pandemic has led to the urgency to develop a safe and effective coronavirus vaccine to combat the spread of COVID-19. Thus, the mammalian downstream technology provides an effective approach for the development of the COVID-19 vaccines. Companies and researchers over the world are looking for ways to address the challenges of this virus, to mitigate the spread and develop a cure for this disease. Many researchers find new drugs and medicines for the cure, along with some computer science researchers focusing on detecting the infectious patients through medical image processing like X-rays and CT scans. Over the years, many advances have been made in bioprocessing as bio-manufacturers strive to increase yield, improve product recovery, enhance product purity. Cells isolated from animal tissues can be expanded in culture for use as a research tool, for the production of virus vaccines and various therapeutic proteins, and to generate functional cells or tissue analogues for regenerative medicine. Mammalian cells can be made to produce vaccines through viral infection, and therapeutic proteins through genetic engineering. Mammalian cell culture is at the core of biomanufacturing therapeutic and viral vaccines. Vaccines work to prepare an immune system to fight off disease by generating an immune response to disease-causing agents, most effective tool to prevent spread of infectious diseases. Hi there. Did you know there is a vaccine that has been successfully produced to tackle COVID-19? I'm sure there are people who still do not know about the vaccine, so I will explain about the vaccine and how it works against COVID-19. The vaccine that has been produced to tackle COVID-19 is Pfizer-BioNTech vaccine. This vaccine uses messenger RNA, or called mRNA, genetic material that our cells read, to make proteins. After injection, the vaccine particles bump into cells and fuse to them, releasing mRNA. The cell's molecules read its sequence, and build spike proteins. The vaccinated cells break up the proteins into fragments that present on their surface. These spike protein fragments can then be recognized by the immune system. When other cells called helper T cells detect these fragments, the helper T cells can raise the alarm, and help marshal other immune cells to fight the infection. Other immune cells, called B cells, may be able to lock onto the spike proteins. If these B cells are then activated by helper T cells, they will start to proliferate, and pour out antibodies that target the spike protein. The antibodies can latch onto coronavirus spikes, then mark the virus for destruction, and thus prevent infection by blocking the spikes from attaching to other cells. The antigen-presenting cells can also activate another type of immune cell, called a killer T cell, to seek out and destroy any coronavirus-infected cells that display the spike protein fragments on their surfaces. The immune system also contains special cells, called memory B cells and memory T cells, that might retain information about the coronavirus for years or even decades, and hence, will remember how to fight the virus if we are infected in the future. Now, let's discuss the differences between cell culture-based vaccines and egg-based vaccines. First, let's discuss about egg-based vaccines. Egg-based vaccines are produced from embryonic chicken eggs. It is the most widely used method for vaccine production that has been applied for 60 years. Chicken eggs are the main source for flu or influenza vaccines that can generate up to 500 million doses. It is produced in forms of inactivated or killed vaccines for flu shot, and live attenuated or weakened vaccines for nasal spray flu vaccine. However, the egg-based vaccine poses a number of concerns, such as insufficient supply, especially in case of epidemics and pandemics, and potential allergic to the eggs. Hence, cell culture technology has been introduced as an innovative approach to overcome the limitations of egg-based vaccines. Cell culture-based vaccines use mammalian cells. The viruses cultivated in cell cultures have higher similarity with the circulating viral strains, hence it offers better protection than egg-based vaccines. Cell culture technology is more efficient, and highly scalable in cases of rapid pandemic response, with a faster startup of the vaccine manufacturing process. That's it for this video. We hope you enjoy it, and thank you for watching.